Hello friends, in this video lecture we will discuss about plant tissue culture. So first about the definition as this is given here. Plant tissue culture is a technique of maintaining and growing plant cells or tissues or organs on an artificial medium in suitable containers in the presence of controlled environmental conditions. Why there is need of tissue culture? If we discuss about the questions, so we already know the traditional plant breeding techniques is not capable to fulfill the demand which is generated by the current growing population or by population explosion. So tissue culture comes into existence, and by tissue culture in a very short period of time we are capable to create thousands or more than thousands plant. G. Haberland is considered as father of tissue culture. In tissue culture program, we can take any part of the plant that is living, and this plant part which is taken for tissue culture program is known as explant. And explant is carrying capability to generate whole of the plant under sterile condition in specific nutrient media. This process is called cellular totipotency. By the year 1950, this was first time identified that by the use of explant, we are capable to create the whole plant under sterile condition, that is cellular totipotency. So what is totipotency? If from a single cell there is capability or possibility to develop whole of the organism, this is known as totipotency. Now here we discuss, we have to grow this explant in a nutrient media for conversion into whole plant. So what is nutrient media? Nutrient media is the sum of a number of substances that is helpful for growth or culture of that particular explant. So here in nutrient media there is presence of inorganic salts. Both micro and macro essential elements that are nearly 17 are needed for growth of explant. Next, as a source of energy or carbon source, most probably sucrose is needed here. There is also addition of a number of vitamins, essential amino acids and growth regulators. In form of growth regulators, there is need of auxin and cytokinin. Auxin is taken mainly in form of 2,4-D, that is 2,4-dichlorophenoxyacetic acid. This form of auxin is very helpful for initiation of root in that particular explant. While BAP, benzyl aminopurine, is taken as a cytokinin, and this causes very quick shoot initiation. So these both phytohormones or plant growth regulators also needed in nutrient media. There is also need of other compounds like coconut milk, malt and yeast extract. They will also help to grow that particular explant. Agar, that is a type of carbohydrate, is also used as a gelling agent. This is obtained from red algae like Geladium and Gracilaria. They also commonly known as seaweeds. So agar is used if we are trying to convert the nutrient media into semi-solid or solid. Before tissue culture program, we have to be very sure about sterilization of both nutrient media as well as explant. What is here about sterilization? So this is about killing or removal of any kind of infectant or pathogen from nutrient media or explant. For explant, that is living substance, we have to use sodium hypochlorite, this also known as Clorox, that is a type of disinfectant. While for nutrient media and apparatus which is used for tissue culture, we have to use autoclave at 120 degrees C for 15 to 30 minutes. Now micropropagation. So micropropagation is also a type of tissue culture, and in this technique we used rapid vegetative multiplication of ornamental and fruit trees by using a small sized explant. So micropropagation is, we can say, an advanced type of plant tissue culture where 1,000 or more than 1,000 plantlets can be created in very short period of time. Each of these plantlets will be genetically identical to original plant from where they are grown, and they all are known as somaclones because they are originated from the same somatic cells without any use of meiosis as well as fertilization. So there is no any possibility of change in their genetic composition. So this process also known as somaclonal propagation, while the resultant is somaclones. Many important food plants like tomato, banana, apple, etc. are produced on commercial scale by using this micropropagation or somaclonal propagation method. Next is about somatic hybridization and protoplast culture. Somatic hybridization involves fusion of two somatic cells of two varieties or species. In flowering plant, only this is stamen and pistil that are respectively male and female reproductive structures present in flower, except that rest of the plant part is carrying somatic or vegetative or non-reproductive cells. So any of two somatic cells from two different varieties or two species are taken out and they are getting fused by a number of methodology. So resultant is called somatic hybrid, and this process is known as somatic hybridization. By using this technique, pomato-like plant, that is the fusion of potato plus tomato. Pomato, same there is bromato, that is fusion of two different cells from brinjal as well as tomato. Now, what is the methodology? So here, first of all, we have to take two different cells. Like for example, this cell is taken from potato and this cell is taken from tomato. They both are individual cells. And here, as we know, around each plant cell, there is cell wall, that is just around cell membrane. So both cells are same, means this is from potato side, this is from tomato side, but cellular composition is almost same, means they are carrying nucleus with cytoplasm and all other cell organelles. So first of all, we have to dissolve their cell wall by using two enzymes, cellulase and pectinase, because these two are respectively responsible for digestion of cellulose and pectin that is present in cell wall. So after digestion of pectin and cellulose that is present in cell wall, only cell membrane including cytoplasm is left behind. So this structure, that is cell membrane with rest of the cytoplasmic cell organelles, is known as protoplast. So here now we obtained protoplast from both sides. This is protoplast of potato, this is protoplast of tomato. Now both these protoplasts are getting fused in an artificial medium by the use of polyethylene glycol, that is PEG, or with the help of electric current that causes dissolution of their cell membrane, and that's why fusion of cytoplasm will take place. So here in this case, both cytoplasmic fusion is going on. Now here cytoplasm is fully fused in this condition, but their nuclei are still isolated. So fusion of cell is known as protoplastic fusion or somatic hybridization. So here this cell is carrying heterokaryon condition. There are two nucleus from different cell along with two different type of cytoplasm. Now there may be two options. So both these nuclei may get fused and get converted into a single nucleus. This structure, this whole structure is known as somatic hybrid. Most of the time both these cells that we earlier taken from here are diploid, so 2n plus 2n conditions may be there. Both these conditions here, both are diploid cells or maybe also polyploid in nature, because polyploid is very common property of plant cells. So here there is fusion of both cell cytoplasm as well as nucleus, and this structure is somatic hybrid, while process we earlier discussed is somatic hybridization. Now there may be one option here, somehow out of these two nuclei, one nuclei is getting disintegrated. So only one nuclei left behind in two different cytoplasm, means cytoplasm of potato and tomato carrying only one nucleus. This may be either from potato side or from tomato side. This cell is known as Cybrid or cytoplasmic hybrid. So this one is carrying cytoplasm. So this Cybrid is carrying cytoplasm of two different cells but only one nucleus, while somatic hybrid carrying two different nucleus as well as cytoplasm from two different cells. By this method, the resultant is termed as pomato, which is carrying characteristics of both tomato plus potato. Now, what are the applications of plant tissue culture? So plant tissue culture is used for a number of purposes that are given here. First is about to generate or to create somaclonal variants. Somaclones, they are carrying genetic variations which develop spontaneously in culture due to stressful conditions, like there may be improper nutrient medium. This causes development of variations in that particular somaclones. So we already know somaclones, these are somatic cells. After vegetative reproduction, these are somatic cells which are getting multiplied by micropropagation or tissue culture methodology. Somaclonal variation is very common when the X plant is taken from non-meristematic regions. Meristematic region in plant is of three types: apical meristem, intercalary meristem and lateral meristem. So if X plant, that is the plant part which is taken for tissue culture program, if not taken from meristematic region, then there is maximum chances of such kind of variations, and these variations sometimes may be beneficial for us, and that's why this also helps in improving productivity. Second, transgenic plants. So first, what is transgene? These are the genes which are transferred into an organism that is not actually related from that one. So suppose there are two organisms A and B, and if inside B we are also transferring gene of A, the particular gene of B is known as transgene, while the organism B is known as transgenic organism. So by this method we can produce transgenic organism, and by the use of micropropagation or tissue culture we can produce bulk of a plant. Third is about virus-free plant. In case of plant, both apical and axillary meristem, they are free from viruses even if the whole plant is infected by viruses. This is because of high content of auxin and their rapid meristematic activities which are going in apical and axillary meristematic region. So by using apical or axillary meristems, we are capable to create virus-free plant. Next is embryo rescue. What is embryo rescue? So here we are just trying to protect or save embryo which is present inside the seed and not capable to survive. So this embryo can be taken out, and by the use of tissue culture we are capable to form new plant from the same embryo. Fifth is induction and selection of mutations. So by tissue culture program we are capable to create mutations by the use of chemical mutagens that will be added in culture medium, or we can expose the whole culture for physical mutagens, and this causes induced mutation, and these mutations may also be sometime favorable for us, so this may also be applicable. What are chemical mutagens? These are substances like alkylating agents, azides, and physical mutagens are a number of rays like gamma rays, X-rays, UV rays. So they may also helpful to induce mutation, and mutations can be continued in next generation if they are favorable by plant tissue culture technology. The next two applications are resistance to weedicides as well as rapid clonal propagation. So we earlier discussed there will be production of thousands or more than thousands plant by using the same micropropagation or tissue culture programs.